Thank you again. As much as I would have liked to be in Paris, that is not possible at the moment. My name is... Everyone hear me okay? GUASTI. And I work in London at the Center for Endocrinology, which is part of Queen Mary University. And basically, I'm going to give you an overview of what has been achieved in terms of reprogramming strategies to obtain adrenal-like cells for not only disease modeling in a personalized manner, but also for the development of novel cell-based therapies. To then move on to gene therapy. So in this schematic, I'll give you an overview of the current strategies for cell-based therapies. And as you can see in the right-hand side, many groups have been trying to generate adrenocortical-like cells starting from different cell sources. And I can tell you that we are lagging a bit behind other fields or even other endocrine fields if you consider the successes in generating functional pancreatic beta cells, which are already in phase 1 and 2 clinical trials for type 1 diabetes. So we still don't have a proper protocol to differentiate embryonic stem cells or inducible pluripotent stem cells to adrenocortical-like cells. But we've had some success by using the so-called lineage conversion or transdifferentiation, which entails the forced expression of lineage determining transcription factors in order to obtain cells of a given lineage. One could also envisage to start from somatic cells directly from the patients, which, for example, congenital adrenal hyperplasia patient, or this is highly studied in my department, familial glucocorticoid deficiency patients, and apply genome editing for correcting the mutations before reprogramming strategies. And of course, once these cells have been generated, they can be used for disease modeling, like we have done. Organoids can be generated. We are not there yet. And they can also be tested in preclinical models of adrenal insufficiency via transplantation with or without the aid of an encapsulation device. So this was our study, which was completed a couple of years ago, where we generated the so-called human-induced steroidogenic cells starting from different sources in humans, from skin fibroblasts, blood, and urine-derived cells. And these cells, through reprogramming, were converted to phenotype.
Pas non plus. Putain. Je le réécris.
Sorry, I can hear. I wonder whether I can. These human-induced steroidogenic cells were used for both disease modeling and in preliminary transplantation experiment to check viability. So in a nutshell, this is our protocol to generate such cells. We have to force the expression of steroidogenic factor at quite high levels with a multiplicity of infection of 200 in the presence of this compound, bromocyclic AMP, which activates protein kinase A pathway, and in the presence of LHRH, which we saw it incremented the fitness and viability of reprogrammed cells. Now, going quickly through the results, the reprogrammed cells, regardless of the source, fibroblast, urine, or blood-derived, they all de novo express an array of steroidogenic enzyme at the mRNA and protein levels. We could never see the expression of sulfotransferase for some unknown reason. And of course, SF1, which is the one that was delivered through lentiviral infection. And by using mass spectrometry, we could assess simultaneously a panel of the steroids. And as you can see here in reprogrammed cells, we have the de novo expression of many steroids, except the HES sulfate, probably because of the absence of the sulfotransferase that we've seen previously. These reprogrammed cells also de novo expressed the ACTH receptor, MC2R, and the associated protein, MRAP. And they responded to both physiological stimuli such as the CTH and pharmacological such as bromocyclic AMP. So this was basically in vivo synactin test. So we went on and started collecting mostly urine from the clinic, from patients with congenital adrenal hyperplasia. We were doing that weekly, and we were isolating the so-called urine-derived stem cells, which were cultured, characterized, and then reprogrammed to steroidogenic-like cells. And then we measure the steroid profile through mass spectrometry. And this is an example of a patient with CYP21A2 mutation, one of the most common, at least missense mutation, resulting in a simple virilizing form of CH with a residual enzymatic activity between 1 and 10%. As you can see here, by comparing the steroid profile between this patient and those cells reprogrammed from healthy patients, you can see an accumulation of metabolites prior to the 21-hydroxylase activity, such as 17-hydroxyprogesterone and pregnenolone, progesterone, and so on, and a downregulation of those after the activity of CYP21A2, such as DOC, cortisol, and so on. So it was phenocopying quite accurately the steroid profile that you would expect by assessing the serum of a patient, of that patient at diagnosis. So we went on and tested the possibility that delivering the wild-type version of the defective enzyme through lentiviral delivery could restore steroidogenesis in these reprogrammed cells. And this was found to be the case in a multiplicity of infection-dependent manner. So more CYP21A2 in this case was delivered through a lentivirus, the higher the effect. Here you have an increase of the cortisol level to levels which were similar to those of healthy cells, and a concomitant decrease in 17-hydroxyprogesterone, pregnenolone, and testosterone to restore steroidogenesis, at least in a dish. And we went on by analyzing cells from other patients with other CYP21A2 mutation, STAR, HSD3B2, and CYP11A1 side chain cleavage mutation. And again, we could restore normal levels of the cortisol by expressing the wild-type version of the defective enzyme. And of course, if you have a CYP21A2 patient cells and you overexpress STAR, nothing happens. Of course, you need to replace the correct gene. So we have generated these cells from different sources, so from skin, urine, and blood, and they acquire ultrastructural feature of the steroid-producing cells. They secrete steroid, they express steroidogenic enzymes, and we found that we can mimic or phenocopy genetic condition or model genetic conditions quite accurately, at least for congenital adrenal hyperplasia, which is the model we chose. And interventions can also be tested in a dish. We are also aware that these cells are far from being applicable in clinical settings. First of all, we always found our cells to be proliferation arrested, and this is something to be expected somehow because we are forcing the expression of SF1 inducing terminal differentiation, and therefore we could never find a subpopulation of these cells with stem progenitor cell characteristic, which should continuously replaced older cells like it happens in the adrenal cortex, which, as some of you might know, is an organ that undergoes continuous turnover. And this is, as I said, probably because of this force, we are bypassing lots of important events which would normally take place at the boundary between the capsule and the subcapsular region, where early differentiation events take place before SF1 starts to be expressed. And of course, we're using lentiviruses, which is not really something that is desirable in clinical settings. So others have attempted to use different sources for cell therapy. For example, some have used bovine adrenocortical cells to test their use in rodent models of adrenal insufficiency, which is normally achieved by performing double adrenalectomy. And these cells can be implanted into either immune deficient recipient or not, if we can use an encapsulation device which would create this sort of immune privilege site where cells can still exchange oxygen and nutrients, but they would not be recognized by other B or T cells from the host. This is such an example, a study done in Dresden, Germany, where they used bovine adrenocortical cells, which were extracted from the adrenal, and they were encapsulated in alginate. Alginate is one of the encapsulation devices which are being used, and it has also a good immunosuppressive property. And I'm just showing you this image here. These are slabs of alginate loaded with adrenocortical cells. And despite the number of animals used is not very high, they using this encapsulation device, this alginate slabs loaded with bovine adrenocortical cells in rats which had undergone double adrenalectomy at day 0, they were able to avoid death of this animal, which normally occurs between 10 days and 2 weeks because of adrenal insufficiency. So this is something that is being researched from a point of view of xenotransplantation. I've also included this part where, in theory, it would be possible to isolate different population of the adrenal cortex. For example, we know of the existence and the phenotype of the cells with stem progenitor characteristics. However, in order to isolate these cells to then use them for either generating organoids or for clinical applications, one would need to determine the expression of at least one cell surface receptors in these progenitor cells. And this is something that we have been doing in the lab for quite some time, because we've been interested in the role of this notch-atypical ligand, which is called DLK1, with an imprinted gene. Its function is known to strongly inhibit differentiation. And is also a bona fide marker of stem cells in the liver of hepatoblast, and also cancer stem cells in hepatoblastoma. We still have a bit of controversy regarding the molecular identity of the DLK1 receptors, because DLK1 itself lacks some key amino acids which are known to be important for its interaction with Notch receptor, even if there are some indication that it might bind to Notch receptor. But we know nothing about the pathway in the adrenal cortex, and this is something that we are currently doing. DLK1 is highly expressed in the medulla. We don't know what it does. And in the cortex, this is a rat, is expressed in a classical, continuous and layer manner underneath the capsule during embryogenesis in the adult, and is expressed together with Sony hedgehog, which is an important self-morphogen, and Sony hedgehog mark progenitor cells in the adrenal cortex, at least in mice. So these are experiments in rats, so it is, we can say that potentially DLK1 cells are also a progenitor in rats. What happens in humans? The expression of DLK1 is similar to that of rats during embryogenesis and postnatally up to roughly the third decade of life, where we see that the cortex remodels to generate these clusters of cells expressing DLK1, which we named DLK1 cell clusters. They increase with size and in number with age, replacing the classical layer and continuous expression pattern. And they are different entities to another cluster structure which has been described, the so-called APCC, or aldosterone producing cell clusters, cluster highly expressing aldosterone synthase and secreting aldosterone potentially in a renin-independent manner. And our DLK1 cell cluster were always found to be different cellular entities compared to APCC. And also considering the phenotype of this DLK1 cell cluster, they are unable to secrete aldosterone or cortisol, and they express other steroidogenic enzyme at a varying level, and they are low SF1 expressing cells. So they can be considered somehow partially undifferentiated cells, but are they really steroidogenic precursor? In order to do so, we generated this mouse model where the fate of DLK1 expressing cells can be traced by using this DLK1 CREAR-T2 ROSA26, and cells, when given to tamoxifen, DLK1 expressing cells will express this red fluorescent protein, and also the progeny of these cells will also be labeled indefinitely. And just a slide of data, this experiment can be done during embryogenesis or postnatally. For example, here tamoxifen was given pregnant dams at E12.5 and adrenals collected at postnatal day 10 and 38. So normally you would have DLK1 cells to be in the periphery of the cortex. However, P10, so we are 18, 19 days after injection, you can see a large number of cells which have infiltrated the cortex. Also at the P38, this kind of column-like cells which have become steroidogenic cells because these are SF1 positive cells. So DLK1 cells are in the capsule, subcapsular region, and they can differentiate to generate steroidogenic cells. The same in the adults, however, they are mostly dormant because even if we do very long chase, we can only see that they participate to roughly 1 to 5% of steroidogenic cells during adrenocortical self-renewal in the adult. However, they can be reactivated in case of need. For example, if you treat mice with dexamethasone, like in human, you determine zona fasciculata atrophy because of ACTH absent. And if you remove if you cease the treatment with dexamethasone, most of the cases you would recover and you would regenerate the zona fasciculata. And again, using this mouse model, we could see that RFP, this is an immunohistochemistry instead of fluorescent. After regeneration of the zona fasciculata, we can see plenty of cells which were once in the capsule and which were once DLK1 positive cells. So we have somehow determined a population or a subpopulation of adrenocortical precursor which express this cell surface protein called DLK1 MB cells are mainly active during embryogenesis, but can be reactivated postnatal if needed. And what it is important here is that these cells can be very efficiently isolated because we have excellent antibodies to DLK1. So now we are collecting these progenitor cells from either mice or human adrenals, and we are attempting to generate organoids starting from single DLK1 positive cells. So lastly, I would like to present you with some data, which is really not ours on gene therapy, just for completeness, but maybe you'll see at the end that even the DLK1 data might converge and help planning some new strategies in gene therapy. So first of all, there are no human trials in the pipeline, but we have some good mouse models of that can phenocopy congenital adrenal hyperplasia. We have CYP21A2 deficient mouse, which is actually a CYP21A1 deficient because the bioactive gene in mice is CYP21A1, while the pseudogene is CYP21A2P. And these mice have, as expected, perinatal mortality, which can be avoided by giving dexamethasone in drinking water. They have high level of progesterone, for example, high levels of ACTH, and low levels of corticosterone and aldosterone. Of course, we cannot mimic the hyperandrogenisms in human in these mice because due to the lack of CYP17, they do not produce androgens. However, there have been a good model since 1999 to test gene therapy approaches. More recently, in our department, we have generated an MRAP knockout mouse, so melanocortin receptor associated protein, whose mutation in human caused familial glucocorticoid deficiency type 2. And so where we have glucocorticoid deficiency and normal renin, angiotensin, aldosterone axis. So this is another model that we would like to use to test either cell or gene therapy. So fast forward to 2018, all the studies, there are like 4 of them which have tested gene therapy for congenital adrenal hyperplasia, were able actually to restore normal steroidogenesis, but this effect has always been transient. And with this more recent study, we know the reason. First of all, the delivery of the transgene, which in this case is wild type 21-hydroxylase, was always done through AAV, adeno-associated viruses. Here we're using 2 serotypes, RH10 and RH9, and injection in the single injection in the tail of these mice. So as I said earlier, high progesterone after the injection, as you can see here in green, the level of progesterone dropped to levels comparable to those of the wild type. Same for ACTH, not quite at the level of the wild type. We have a decrease of pituitary ACTH in the serum, suggesting that some restoration of the steroidogenesis has taken place. However, as you can see, as weeks go by, you can see that progesterone rises again to levels which are back to those of the untreated mice by week 10, and the same for ACTH a bit later on. But what they did here that is interesting is that they assessed the cell type which were efficiently transduced by these adenoviruses. And as you can see here, we have a very efficient transduction efficiency of this AAV in the zona fasciculata. Medulla is negative, zona fasciculata is positive. This is 2 weeks after a single injection. But as time goes by, you can see the number of transduced cells in the zona fasciculata is decreased at 6 weeks, a few remaining at 16 weeks, and nothing left at 32 weeks, which is concomitant to the restoration of a complete CH phenotype in these mice. And if you look in this panel here, you can see that both the capsule and the subcapsular region, the zona glomerulosa, is completely unaffected, untransduced by these adenoviruses. And as I mentioned earlier, we know quite a few things now related to adrenal biology. We know that the location of the stem cell population is within the capsule or within the subcapsular region. And these cells, through potentially asymmetric division, give rise to other stem cells, but also cells which are able to differentiate to functional zona glomerulosa, zona fasciculata, zona reticularis. And through centripetal migration of the older cells, this will eventually die of apoptosis at the boundary with the medulla. So we have cells with stem cell properties in here, and new cells which are generated become functional steroidogenic cells, and they migrate in a centripetal manner. And in 6 months, in mice at least, this process is complete. And you can understand that if these cells here are not efficiently transduced by AAV of these serotypes here or other delivery strategies, the amelioration of the phenotype can be only transient, because new cells are generated which will be mutant in case of humans, or CYP21A2 deficient in case of mice here, which will replace the ones that were transduced initially. So here it's kind of important in the future that we are able to target more the adrenocortical stem progenitor cell compartment for gene therapy, for example. And now we don't have any kind of data to present yet, but because we are able to isolate, for example, DLK1 positive cells from the rest in humans, we are testing different serotypes of AAV or different delivery strategies to assess, to find some that are able to efficiently deliver transgenes and genetic materials specifically to stem cells. So broad conclusion, we can say that cell and gene-based therapies approaches to tackle adrenal congenital disorder are moving at a reasonable pace. I might say, of course, not as fast compared to other conditions. For example, type 1 diabetes. There are like dozens of high caliber groups working on cell replacement strategies for type 1 diabetes. They have excellent protocols to derive beta cells from embryonic stem cells. So we are roughly maybe a decade behind them, and mostly because there are not many groups working on adrenal in terms of cell and gene-based therapies. However, we can follow and learn from them, especially the encapsulation devices which are currently using clinical trials for type 1 diabetes can also be applicable to cell-based therapy for congenital adrenal disorders, and also some reprogramming strategies. So we have to closely keep an eye on what's being done in other fields and try to apply those to adrenal disorders. So thank you so much, and I would like to also acknowledge the work done by members of my group, especially the reprogramming was done by Gerard Ruiz-Babot, who is now in Boston, and is actually continuing to work on this field, and is trying really to establish adrenocortical-like cells from either embryonic stem cells and inducible pluripotent stem cells without the need of overexpressing transcription factors. So something that would be more amenable to be used in clinical settings. Thank you so much.